Hey everybody, welcome to week 5. This week we're going to just talk about anaerobes. So one PowerPoint, which makes it nice. Let me just share so we can get it popped up here and start. All right, so again, anaerobes, they do not grow in oxygen, so they have to grow in obviously anaerobic conditions, basically meaning without oxygen. There's a lot of different ways that you can create this. Common ways would be one, having an anaerobic incubator. So what they call an anaerobe chamber that you can just put it in if you do tend to work up a lot of anaerobes. My lab, when I worked micro, didn't work up a ton of anaerobes. I mean, we would always set up wound cultures. We'd put one in an anaerobic, what we call bag or pouch, in case there was an anaerobe causing that wound infection. Then if it was, we sent it out. We didn't work it up. We were a smaller micro. So a lot of times, not every micro lab will work up anaerobes just because it depends on how much they see it. So in that case, a lot of times they use an anaerobe pouch or an anaerobe jar, and they create an anaerobic environment in there with a CO2 generator. Otherwise, if you're a larger lab that commonly works with anaerobes, you probably have an entire anaerobe incubator. For specimen collection and processing, not every specimen is going to be great for anaerobic culture. Like, you would not set up a throat swab for anaerobic culture. Anaerobes are not going to live in a throat with oxygen all around them. Like, it just doesn't work. So, I mean, you have to think about it that way. There is definitely a listing of acceptable, unacceptable specimens for anaerobic culture that you do need to be aware of. So the best ways to obtain anaerobic cultures is biopsies or aspiration with a needle and syringe. Swabs are generally a poor choice, just to keep that in mind. A lot of times anaerobes do make up our normal flora, just like our other type of bacteria. So sometimes the infections happen the same as with other bacteria. Your own normal flora spread to a sterile site or take opportunity of a situation and cause infection. Now again, there's anaerobes, of course, found in the environment. So here are some listing of anaerobes that are normal flora. In the intestines, we have Bacteroides, Clostridium, and Bifidobacterium. Bacteroides and Clostridium are definitely groups that you're going to want to be aware of. Upper respiratory type tract normal flora is Prevotella, Fusobacterium, and Veillonella. And then you do have a little bit of anaerobes on your skin normal flora, Propionibacterium. The big one there with that is a blood culture contaminant because it can be found in the skin. When you go to swab or when you go to collect your blood culture, you have to make sure you don't hit anything on the skin because it can be contaminating it. So we will start with our Clostridium. We're just going to go through the more significant anaerobes. I'm not going to try to teach you them all. We only have 1 week on this, so we're just going to hit the highlights, the big ones. So Clostridium are gram-positive rods, and they do form spores. The only other real gram-positive rod that forms spores that we have learned so far is Bacillus. So Bacillus is aerobic or facultative aerobic, whereas Clostridium is anaerobic. Clostridium botulinum and Clostridium tetani, when they form spores, they tend to appear like tennis rackets. Now, there are different antibiotic discs that we really use to help determine which anaerobe might be present. So think of like when you learned your gram-pos toxi, you learned about bacitracin, which is one type of antibiotic disc. You learned about oxykin, which is another type of antibiotic disc, novabiocin. So kind of similar to that, we can use discs here to see if susceptible or resistant to them. So with Clostridium, they are very much susceptible to kanamycin and vancomycin. There are some specialized agars that you can grow. Egg yolk agar is very commonly used to grow Clostridium. And then there is an even more selective agar called CCFA that is specifically looking for Clostridium difficile. C. diff is a very much something we see in the hospitals, we see in nursing homes, things like that. So we'll start with our first Clostridium. There's going to be 4 that we'll ultimately go through. Clostridium botulinum causes botulism, which is like basically really severe food poisoning. You probably know this because of its Botox, things that people use for Botox. So what it does is it has these neurotoxins that will cause paralysis of your different muscle groups, and they have figured out a way to put that into this form for Botox and inject it in small amounts into different muscle groups to cause that nice freezing look, to smooth out the wrinkles so people look a lot younger. But the problem with this also is if you find the neurotoxin still left behind in the food, it will also cause you extremely severe food poisoning and infant botulism. So it can be seen through maybe formula, baby formula. The toxins are also potentially bioterroristic, to be launched as bioterroristic. So when this grows, it will be beta hemolytic on the agar when it grows. Clostridium tetani causes tetanus, which is commonly known as lockjaw. So the neurotoxin released here will paralyze your muscles, causing that lockjaw effect. A lot of times this comes from a puncture by something that's contaminated, like people often think of stepping on a rusty nail, things like that. That's why it's so important to get your tetanus shot updated every 10 years. This will have more of a narrow zone of beta hemolysis when it grows. Clostridium perfringens causes food poisoning and gas gangrene. Gas gangrene is basically a destruction of your muscle and tissues. There's some amazing pictures of that out there if you want to look those up. These are, this will have what we call a double zone of beta hemolysis, and that is a very big characteristic for Clostridium perfringens, is that double zone of beta hemolysis. Very unique to this one. And this is the gram stain of Clostridium. Overall, Clostridium tends to be large gram-positive rods, just like bacillus, that form spores. You can't really see the spores in them very well, but again, large gram-positive rods. And then here is the double zone for Clostridium perfringens of beta hemolysis. So it shows you zone 1 and zone 2. And then finally, our last Clostridium is Clostridium difficile. We just call it C. diff in the healthcare world. Very much spread nosocomially. That's why you see it a lot with hospitals, nursing homes. Very severe diarrhea. A lot of times this comes about after people have taken a lot of antibiotics. So if they've been on antibiotic therapy and it kind of wipes them out of their normal flora, this one will come up and rise up and take advantage of that and then cause severe diarrhea as a result. It produces two toxins. Toxin A is responsible for that diarrhea disease, and toxin B is just something that we're not sure what it does quite yet, but there are two toxins. So a lot of times labs will just have a kit on hand that they can test the stool for this toxin and determine if somebody's positive or negative that way. A lot of times we do a kit because it's quick, it's easy, and if somebody does have C. difficile in the hospital or nursing home, they need to be put into isolation. We can't have this continuously spreading because it is so quickly spread. So they need to put in isolation so that you have to gown and glove up, that kind of thing when you go see them, so that you're not passing this along to other patients. So that's why a kit is always great to have on hand. It gives you a quick pause or neg right away. Otherwise, when C. diff grows out, again, CCFA is a specific agar for it, and it will be yellow colonies on that agar when it grows. So there is the CCFA and the yellow colonies. All right, Bacteroides fragilis is a gram-negative rod. Pretty much the most common anaerobic infection you will see is by Bacteroides fragilis. This is probably the #1 guy you'll see out of anaerobes. Can basically cause infections throughout the body, but generally it occurs below your diaphragm. Very foul odor associated with this one. I mean, I know a lot of things stink in micro, but extremely foul odor that is seen here. There is a specific agar used to grow this out called BBE, which is Bacteroides bile-esculent agar. Bacteroides loves itself some bile. Oh my gosh. Loves growing in the presence of bile, so this agar is really ideal for it because it will definitely thrive and grow on it. For the antibiotic discs, it is resistant to all three main ones that we test with. So it's resistant to kanamycin, vancomycin, and colistin. Oh, there is one other agar as well, LKV, that you can use too. So again, there's a gram stain. It's a gram-neg rod. Looks like any other gram-neg rod. And so there is where the arrow, the BBE agar, showing you the positive growth of Bacteroides fragilis. That was a split plate with blood agar on one side, BBE on the other, just so you know. All right, our next one is Fusobacterium nucleatum, another gram-neg rod. The key characteristic here is that the rods basically have a pointed or tapered ends. Typically, the infections occur above the diaphragm, or the head, the neck, the pleuropulmonary. Non-hemolytic, this one is sensitive to kanamycin, and it will fluoresce a chartreuse color, which is like a bright neon-y yellow, if you will. So there is a Fusobacterium nucleatum with very pointy ends. I know it's hard to see in a picture, but very much more slender looking than the other one. They kind of look like little needles or something. Prevotella, another ground egg rod. Again, infections occur head, neck, pleuropulmonary. It will fluoresce brick red. It does like bile, so sometimes it'll be bile sensitive, and then it can grow on LKV agar as well with the black pigment. Kanamycin and vancomycin resistant when we use those antibiotic discs. And there it's showing the LKV black pigment on the agar. Porphyromonas, another gram neg rod, same type of infections, also fluoresces brick red. If you remember back to heme, porphyro, remember, was the protoporphyrin ring, which is always red in color. Porphyrias always cause red urine, so porphyro always goes with red, if you will. So that's why it's easier for me to remember it fluoresces brick red. And there's the fluorescence. Isn't that pretty? Looks pretty. Okay, Propionibacterium was the one that is associated with contaminating blood cultures coming off the skin. It is a gram-positive rod, and actually the full name is Propionibacterium acnes. Just like the name applies, this is what's responsible for that inflammatory acne. It causes, acne, basically a skin infection of acne. And then other ones, Peptostreptococcus anaerobius is a gram-positive cocci. This is also has an association with blood cultures in that, and we'll learn this soon here, that the anticoagulant that is in the blood culture bottles will inhibit Peptostreptococcus from growing. So if somebody ever did have a septicemia with Peptostreptococcus, it would not grow in those blood culture bottles to let us know, because that anticoagulant inhibits it. Luckily for us, it's not that common to see. Veillonella is a gram-negative cocci, and then Bifidobacterium is a gram-neg rod. All right, those are your anaerobes. Try to keep the main ones separated in as far as like the test results, the diseases that they cause, and that kind of thing, and you should do just fine. So reach out if you need anything, as always. Otherwise, have a great week. Thanks, guys.